Namaste everyone, welcome to online classes. So today in our biology class we shall be learning about some of the important cell organelles. Okay, so in our previous class we had learnt about one major component of the cell called as nucleus. We had learnt about the differences between the plant cell and the animal cell. We also had seen about prokaryotic and eukaryotic cell, right? Now let us understand about the different cell organelles. We have to mainly concentrate about their structure, functions, and certain highlights of it. Okay, now getting into the first organelle of the day, mitochondria, right? So this mitochondria is also called as the powerhouse of the cell. Okay, why this mitochondria is also called as powerhouse of the cell? Because it helps in the synthesis of the energy in the form of ATP. Okay, always energy is stored in the form of ATP. What is ATP? Adenosine triphosphate. Okay, so the energy is produced by this cell organelle and therefore it is called as powerhouse of the cell. Coming on to the structure of mitochondria, this is also a double membraned structure. The outermost is called as the outer membrane. The inner membrane is folded. Can you see here folded? Right, so it is folded and the folds are called as cristae. Okay, each fold, can you see here? Right, so that inner membrane is folded to form cristae and it will have this stacked particles, that is F0 F1 particles, which helps in production of ATP. Okay, so inside the membrane it contains a jelly like substance called as mitochondrial matrix. Like how we have cytoplasm inside the cell, similarly we have mitochondrial matrix inside the mitochondria. Fine, so it also has got its own DNA and its own ribosomes. Fine, so they can prepare their own proteins. Fine, so they are able to make their own proteins. That is the speciality of the mitochondria. There are only two cell organelles in the cell which has got its own DNA and ribosomes. It is mitochondria and chloroplast. Fine, so this is about the structure and function of mitochondria. Fine, so this mitochondria was invented by a scientist called as Kolliker. Fine, now getting into the next cell organelle called as chloroplast. So in our previous slide we had once come across plastids, right? So these plastids were of three types, chromoplast, leucoplast and chloroplast, right? Under this leucoplast they are starch granules, they store starch. Okay, under chromoplast we have chloroplast. Fine, so chloroplast is green in color because they have chlorophyll. These chlorophyll is prepared by small disc-like structures called as thylakoids. Okay, before that let me explain the structure for you. Chloroplast contain a double membranous structure, the outer membrane and the inner membrane. Within the inner membrane there is a fluid like substance, like how we had in the mitochondria as well, right? That is called as stroma. Okay, there in mitochondria we had mitochondrial matrix, like that we have stroma here. Fine, inside the stroma small disc like structures are present called as thylakoids. These thylakoids are piled up on one another to form grana. Okay, each grana is connected with each other by the help of stroma lamella. Okay, can you see the connections between the granas, right? So they are called as stroma lamellas. Okay, then they have their own DNA called as plastidal DNA and they have their own ribosomes. Fine, as we very well know that they produce chlorophyll, they are called as kitchen of the plant cell because they help in the process called as photosynthesis, that is for the preparation of the food. Fine, now getting into the next uh, cell organelle. It is endoplasmic reticulum. Okay, so where does it start? Can you see here nucleus we have? So the nucleus is also a double membrane structure, the outer membrane and inner membrane. So this endoplasmic reticulum will start from the outer membrane of the nucleus. Okay, it is attached to the outer membrane of the nucleus and it traverses throughout the cell, and therefore it is called as skeleton of the cell. Okay, so it is of two types, called as rough endoplasmic reticulum and smooth endoplasmic reticulum. It is called as rough endoplasmic reticulum because it contains ribosomes on it. Can you see your red color dots on the endoplasmic reticulum? So this is called as rough endoplasmic reticulum because they contain ribosomes. Since they have ribosomes, what would be the function of them? It is the synthesis of proteins. Okay, and there is another type called as smooth endoplasmic reticulums towards the outer side of the cell. Okay, so that smooth endoplasmic reticulum will help in the synthesis of the fat molecules, okay, lipid molecules, and these lipid molecules are very essential for the manufacture of the phospholipids that are required for the cell membrane. Okay, therefore the process of synthesis of the cell membrane from the lipids is known as membrane biogenesis. So the endoplasmic reticulum is also involved in the process of membrane biogenesis. Okay, so these are the major things that you have to concentrate in endoplasmic reticulum. Fine. Now getting into the next organelle called as Golgi apparatus. Okay, so since it was invented by Camillo Golgi, scientist called as Camillo Golgi, it is named after him, Golgi apparatus. We can also call it as Golgi complex. Okay, now this Golgi complex will contain series of tube-like structures. Can you see here? They are called as tubules, and small sac-like structures, can you see here? They are called as vesicles. Okay, so whatever it might be, proteins, it might be enzymes, it can be anything that is synthesized by the cell, will be packed by the help of vesicles and tubules here, and it is sent outside the cell, or it is sent within the cell wherever it is required. Therefore they are called as export houses of the cell. Fine, they are very well versed in the packing of materials. Fine, so the tube like structures are called as cristae, or it is called as cisternae, and the ball like structures are called as vesicles. Okay, so this is about Golgi apparatus. Fine, now getting into the next structure called as lysosomes. Lys means what? Lys means to break. Okay, so those organisms which help in breaking the unwanted toxic substances that is present in the cell is called as lysosomes. Okay, they're called as suicidal bags. They are also called as scavengers of the cells. Okay, so why they are called as scavengers of the cell? Because just now I told you they help in removing of unwanted substances, right? And sometimes what will happen, if the cell gets damaged anywhere, that's the whole cell gets bursted out, it gets engulfed by the lysosome, right? Therefore it clears the body like that. Fine, therefore they are called as suicidal bags of the cell. Fine, how can they remove the toxic substances? How can they digest the enzymes, something like that? That is because of the presence of the strong enzymes called as hydrolytic enzymes in them. Okay, so these enzymes will be released to wherever area they are present to remove the worn out cells or old cells or damaged cells. Or sometimes what will happen, if the bacterial attack has taken place to the cell, it has to be thrown out, worn out, right? So then in such cases also lysosomes will help in a larger extent. Fine, so this is the activity of lysosomes. Getting into the cell membrane, we had told in our previous class while doing the cell membrane The cell membrane is a semi-permeable membrane. That means it can allow only certain substances to pass through them. It will not allow all the substances to pass through them, right? So based on this property of a cell membrane, we have two processes called as diffusion and osmosis. Okay, so movement of particles, how does the movement of particles takes place inside the cell? So that is very much well explained here. So diffusion, the process of movement of molecules from a region of higher concentration to a region of lower concentration is called as diffusion. Okay, especially here the gas molecules will be exchanged. For example, carbon dioxide and oxygen will be exchanged into the gases by the process of diffusion. Okay, so this is about diffusion. Then coming on to osmosis. Osmosis is a process in which the molecules or the substances move from a region of its higher concentration to a region of its lower concentration across a semi-permeable membrane. Okay, across a semi-permeable membrane, that is across the plasma membrane, is called as osmosis. Okay, so various materials like certain solute molecules, all these solvent molecules, water, etc., will be exchanged by the process of osmosis. Okay, so this is about the two processes, how the exchange of molecules take place. Fine. Now getting into what will happen if we put an animal cell or the plant cell into a solution of sugar or salt water? Let us understand this first. Fine. So what is a solution? Solution is a mixture of solvent and solute. Solute is a solid substance, solvent is the liquid molecules. Okay, so there are three types of solutions we have: hypertonic solution, hypotonic solution, and isotonic solution. Hypertonic solution means what? The concentration of solute is more outside the cell than inside the cell. Okay. Hypotonic solution means what? The concentration of the solute is less outside the cell than inside the cell. Okay. Isotonic solution means what? Both the concentration of the solute both inside and outside remains constant. Okay. So this is about solutions, three different types of solutions. So if we put an animal cell inside the hypertonic solution, what will happen? The animal cell, as we told, the concentration in hypertonic solution, the solutes will be more outside, and inside the cell it will be less. So what will happen? As we know, because of osmosis, the solutes from higher concentration will move into the lower concentration, right? So the plasmolysis, plasma membrane will blast. Okay, plasmolysis will take place. The movement of molecules, it will take place outside, that is the solvent will be less, right? Therefore what will happen? From inside the cell, the solvent molecules will get outside into the cell, and therefore it will shrink. The cell will shrink. Okay, and then it can blast also. That is called as plasmolysis. In hypotonic solution, what I told you, the concentration of solutes will be less outside and more inside. Therefore what will happen to the solvent? Solvent will be more outside and solvent inside will be less. Therefore what will happen? The water molecules from outside, it will start entering into the cell. Okay, so therefore it will swell itself. The cell will swell and become bulged, and if it is left as such for a longer duration of time, the cell will also burst. Okay, that is called as cytolysis. Fine. In an isotonic solution, both the solvent and solute remains constant inside and outside the cell. Therefore nothing will happen to the cell. Okay, so but the plant cell, what happens? Plant cell will have cell wall, right? So it might shrink, but it will not blast because it has got the cell wall, which is very very rigid in nature. Okay, so this ends the chapter of the fundamental unit of life. Fine, so we shall continue the further topics in our next class. Thank you.